Hello everyone, this is Mike Yeoman, and today what I'm going to be trying to do is convince you that we should be using bioremediation techniques in the lower Mississippi Valley in order to control methyl parathion contamination that may be occurring there. So the first question that we need to ask is, what is methyl parathion? And it is a very common agricultural pesticide. It is used to control insect populations, and the way that it does that is by destroying the nervous system of those insects. Now that's fine if the target organism is actually an insect. However, it behaves the same way on humans and aquatic invertebrates, shutting down the nervous systems of those organisms as well. And those symptoms may look anywhere from like a headache all the way to death of those organisms depending on the concentration. The individuals who are at most risk of exposure are agricultural workers, obviously because the fields are being sprayed with the chemical, and if they go in too soon afterwards, or if the rains wash some of that pesticide down, they could be exposed to that. There are some other situations where you could be exposed to methyl parathion, but that is usually a result of inappropriate use, such as people spraying their homes with methyl parathion, knowing that it's an insecticide, they're trying to kill the insects, but don't realize that there are problems with using that around themselves and children, and so there has been some poisoning due to inappropriate use. But I'm going to be focusing mainly on the intended use of methyl parathion, which is as an agricultural pesticide. So methyl parathion has a couple different toxicity types. There's an acute toxicity, which is short-term, immediate. That's because methyl parathion can be taken in a number of different ways. You can inhale it, you can ingest it by drinking contaminated water. It can absorb to the skin, especially if there is a rain shortly after the chemical was applied, keeping it dissolved in the water and on the surface of the plant. So then as agricultural workers go into the fields, it just absorbs into their skin. Symptoms may look anything from like a bloody nose, shortness of breath, cramps, all the way to cardiac arrest, respiratory paralysis, or even death. Now, something that is sort of ignored is the chronic toxicity. So what they're finding is that long-term exposure to methyl parathion can result in impaired memory, depression, insomnia, a variety of other types of neurological effects that do occur over long periods of exposure. Part of the reason why there is so much exposure to this is because, as I already mentioned, methyl parathion is very soluble in water, so it can be transmitted very easily. It can move around, especially if it rains shortly after being sprayed. It also has a very low vapor pressure, so it can end up in the air and be breathed in very easily, as well as if there are droplets of water that contain methyl parathion, that can be inhaled and cause some respiratory distress. There are a number of different metabolites as methyl parathion is broken down in us by the liver. But the main metabolite that I'm going to be focusing on is paranitrophenol. That is kind of the end result for humans, but it can still be toxic towards other organisms as well. So like I said, methyl parathion has a number of different symptoms, and a lot of it depends on the mode of exposure. So if you're getting it to the central nervous system, if you're getting enough of it to get to the brain, the spinal cord, by either inhaling it or consuming large amounts, then you might see a headache all the way to coma or death. Other types of exposure, like through the skin, which is a little bit more moderate, you might just get something like cramps, indigestion, vomiting, some visual disorders if you were to say rinse your eyes with some contaminated water. So a variety of different types of symptoms, and I would say they're all serious depending on the amount of exposure that you have. So as we said, methyl parathion is used for agricultural purposes, typically on row crops, and so I have a few examples here. We have soybeans down here at the bottom. They are used fairly often with soybeans and corn, but cotton is probably the most common crop that methyl parathion is used for. It's actually sometimes referred to as cotton poison because of its association with cotton plants. Now, looking at methylparathion, it is an organophosphate, and all organophosphates have a similar structure, and that similar structure really focuses on this pentavalent phosphorus at the center of this molecule. They all have that. I'll show you some examples here in a second. And what they do is they interfere with the acetylcholine esterase enzyme, which reuptakes acetylcholine. And if you think back to your biology classes, acetylcholine is a neurotransmitter, and so if that is not taken up again, if it does not go through that reuptake, then you cannot pass any more messages down that nerve. So interfering with that acetylcholine esterase enzyme prevents any new messages from being passed along the nervous system, or at least that part of the nervous system. Now, we do have enzymes that will break this down, that will detoxify methylparathion and other organophosphates. The concern is how quickly will that be done before it causes that damage. They will detoxify on their own with exposure to light and water through the process of hydrolysis. So up the top here, I have that characteristic pentavalent phosphorus on the methylparathion that exists on all organophosphates, excuse me. And on the left side of the page, you can see that all of these pesticides share that same pentavalent phosphorus there. Very similar structures. Obviously, there is some variation that gives each chemical its own unique behaviors, but they all behave the same way towards that acetylcholine esterase enzyme. Now, on the right side, I have some other organophosphates. However, these are nerve agents that are used in chemical warfare. You may recognize a few of the names like sarin gas or soman or VX gas, but they all share that same pentavalent phosphorus at the center, making it part of that organophosphate family. And they are going to behave a little bit differently, but that structure is still going to interfere with that acetylcholine esterase enzyme. Now, given enough time, given the presence of certain enzymes, they will all break down. They will be metabolized into less toxic, at least for humans, less toxic metabolites. And so the end result is that they are all hydrolyzed. And so this is just showing its behavior, how that pentavalent phosphorus interacts with the acetylcholine esterase enzyme. So you can see that there's a hydroxyl group here, and so it ends up bonding, making that acetylcholine esterase enzyme non-functional until that organophosphate becomes hydrolyzed, which is that end result. So we've talked about what methylparathion is doing to people and where it comes from and where it's used. Now we need to look at where it would be very likely to have this type of contamination. And like I said, it is oftentimes referred to as cotton poison. So I was looking for a location that would have large amounts of cotton, and that brought me to the lower Mississippi Valley, and I chose specifically the northwestern part of Mississippi. And if we look at this upper right-hand corner map, you can see there's an outline of the states. It has the blacked out portion here. And this map is borrowed from the Organic Geochemistry Research Group. They are doing their own project trying to determine the level of methylparathion that is in runoff water in these locations. They're looking at a variety of water sources, but they're just trying to figure out whether or not methylparathion is an issue. So I thought this would be since they're already searching for methylparathion in these places, I thought this would be a perfect place to set up some bioremediation so that whatever methylparathion they do find can be remediated much faster. So I was focusing on this portion right here of northwestern Mississippi. You can see the red dots are some of the site locations that they are trying to work on, and they have some places that have a very large amount of cotton. Overall, this part of Mississippi, about 30% of all the agricultural fields are cotton fields. So very good chance that there's going to be a large amount of methylparathion used. So when trying to determine what types of organisms I would want to be using in this location, I needed to know what soil types were available. And so I pulled up this soil order map of that northwestern Mississippi. And so this is the area that we're looking at, outlined in black. And if you look over to the key on the right-hand side, you see these are primarily vertisols. And vertisols are soils that have a high amount of organic content, usually somewhere between 1 and 6%, but then they also have a large amount of clay involved as well. So there's not necessarily a certain percentage, but a high volume of clay in the soil. Okay, looking at some of the suborders, these are all vertisols that are present. And again, we're looking at this northwestern part of Mississippi, and these are all considered aquarts. And what makes aquarts unique compared to other vertisols is that they have a very high water table. It's either at or very near the surface of the soil. So here are a couple pictures of vertisols and aquarts in particular. So on the right, and this is just kind of a general vertisol that you have some organic content. That's that dark top layer. And then as you get deeper, you can see there's a little bit of a color change here where you're getting into some more clay. So you're getting a lot more clay content in the soil. I think the picture on the left really illustrates this a lot better because you can see that very thick, dark organic layer, and then they have kind of a red clay beneath it. But this also shows you that water table, how close that water table is to the surface of the soil. Now, why this is important is because vertisols behave a little bit differently than some other types of soils because of the high clay content. And so I wanted to include this diagram because it shows how water runoff is affected by the different types of soils. So on the left, we have sand, very large particles, and so water can pretty easily move through these sizable cracks. In the center we have silt, so a little bit smaller particles. Water can still move through, but it's going to be slower. It drains not quite as quickly as sand does. And then over on the right, we have very small particles, very tightly packed, and so water doesn't flow through this very easily. So there's going to be a higher percentage of runoff. So as it rains, any methyl parathion that's going to be absorbed into this water, which could potentially be a lot since it is so water soluble, is going to very likely just run off into other pools. It's not going to seep down into the groundwater, at least not very likely. So we have a location that has potential methyl parathion in it. The way that we've gotten rid of methyl parathion conventionally is surfactants. So basically just soaps. They take soaps, put them on the soil, and then let it absorb, kind of make these little packages of methyl parathion, and then they scoop it up and they take it somewhere else and let it decompose. So they're not really getting rid of it, they're just moving it to another location. And they'll still let the hydrolysis process continue by exposing it to water and light. Not so much with pesticides, but with some of those nerve agents. Another way to get rid of organophosphates is by incinerating them. So that is another process, but they probably wouldn't do that with methyl parathion just because that'd be a lot of work for something that is going to degrade in a matter of a few months on its own anyway. So looking at possible bioremediation, there are a lot of different species that have enzymes that break down methyl parathion, and that enzyme is called methyl parathion hydrolase, so very appropriately named. Some of these organisms behave in slightly different ways. Some of them will sequester methyl parathion or other types of contaminants, and others will hydrolyze the contaminants so that it actually gets broken down into those metabolites. And so I have a number of different microorganisms that will do that down here. And some of them will even go a step further, and they'll use, instead of just breaking it down and trying to remove that methyl parathion because it's a poison, kind of the same way that our liver removes toxins, some of them will actually use the methyl parathion as a carbon, phosphorus, or nitrogen source. So that's kind of an interesting adaptation. They have bioengineered some of these microorganisms to do that intentionally. So because there are so many different organisms that will remediate methyl parathion, we have a very extensive list of possible bioremediation organisms. However, many of the organisms that will break it down are microorganisms, those microflora that occur naturally in the soil. They will break it down. The problem is that metabolite, the paranitrophenol that I mentioned earlier, is more toxic to them than the methyl parathion is. So exposing these organisms to the methyl parathion, which then breaks down to paranitrophenol, that will end up killing at least some of those microorganisms, unless you have some sort of bioengineered version. So wanting to avoid the bioengineering, because then you have risks of exposure of allowing this bioengineered version of a microorganism to possibly reproduce and spread this genetically engineered version around, I wanted to go with something maybe that could be contained a little bit more, so a macrophyte. And so there were two possibilities. There's water hyacinths and cattails. And water hyacinths are an aquatic plant, and they do a great job of removing methyl parathion. And so in one study, after 10 days, 99.9% of the methyl parathion had been removed in the presence of water hyacinths. Now, the scientists who were doing this study recognized that it wasn't all the water hyacinths, so they said that in a controlled group, 40.1% of the methyl parathion had degraded on its own. Excuse me. And looking at the group that had the water hyacinths in it, they determined that 67%, a little bit more than 67% of the degradation was due to the water hyacinths, and about 24% was due to microorganisms that were in the water. So those same microorganisms that we just mentioned. So very, very effective. The alternative are cattails. And cattails are very versatile, and they are very effective at eliminating methyl parathion. Maybe not as effective as the water hyacinths. They only removed 88% of the methyl parathion after 14 days. However, 79% of that removal was in the first 3 days. However, there are some benefits. Cattails already exist in Mississippi. They really exist in all lower 48 states, and they are effective at removing other types of contaminants such as heavy metals, other pollutants. So they have a wide variety of types of bioremediation possibilities. So my suggestion is that we use cattails to remediate the methyl parathion runoff that exists in Mississippi. The reasons for that is that cattails are almost as efficient as water hyacinths, but on the other side of that, cattails are non-invasive, whereas water hyacinths are considered a nuisance in many locations. They build up and they can prevent boat traffic. They're causing problems in many locations because of their uncontrolled growth. In addition, cattails could be used as a biofuel in their locations. Contrary to the water hyacinths, if I wanted to prevent the spread of water hyacinths into other locations, I'd have to build a facility and then have an ex situ type of situation where we're removing the water and cleaning it at a facility and then releasing it. Using the cattails, we can do it in situ, where we can take already existing pools of runoff and plant cattails in those locations, or we could make some artificial wetlands and have cattails in those locations to help clean the methyl parathion in those locations. Cattails are very resilient. They resist the methyl parathion and the paranitrophenol, so it has very, very little downside to filtering out the methyl parathion. So here are my list of citations, and thank you very much for your attention, and I hope that you take my proposal seriously and that we use cattails as a bioremediation technique to control the contamination of methyl parathion. Thank you.